In 1994, beneath a centuries-old church in Hungary, workers broke open a crypt sealed since 1838 and found something no one expected: more than 260 mummified bodies, perfectly preserved, holding secrets that could change how we fight one of humanity's deadliest diseases. What hidden epidemic did these forgotten families face? And why do scientists believe their remains might unlock a cure for tuberculosis today? The answers begin with the shock of that first breath of cool, ancient air. Dust drifted through the air as the crowbar pried loose a slab of crumbling brick. Cold, dry air poured from the darkness, sharper than anything outside. The workers stood frozen at the threshold, blinking into the gloom. Inside, row after row of wooden coffins stretched into the shadows, their painted lids stacked with careful precision. Each box, some barely longer than a child's arm, bore faded flowers, crosses, and curling script in Latin and Hungarian. The silence pressed in, broken only by the scrape of boots on stone. The first flashlight beams caught a glint of gold thread, embroidered silks draped over skeletal hands, rosary beads tangled in delicate fingers, leather boots curled and stiff with age. The bodies themselves, untouched by time, stared back with hollow eyes. Skin clung to bone, hair still braided or tied with ribbons. The air carried a faint scent of resin and old wood, preserved by the crypt's perfect stillness, cool temperatures, steady airflow, and the pine-lined coffins that soaked up moisture and kept decay at bay. Burial records, handwritten and yellowed, sat atop some caskets. Dates ranged from 1731 to the early 1800s. Names and prayers mingled with the artifacts, lockets, holy cards, and scraps of lace. The crypt had become an accidental archive, freezing a century of lives in a single forgotten chamber. Merchants, priests, mothers, and children lay side by side, their last moments marked by the careful hands that dressed them for eternity. Every detail survived: the stitching of a child's bonnet, the faded blue of a ribbon, the worn leather of a tradesman's shoe. The air, still and cold, preserved not only bodies but stories, each one waiting to be read in the hush of the sealed crypt. Tuberculosis did not announce itself in the streets of 18th-century Vac. Its presence seeped quietly into every home, disguised behind parish records that listed causes of death as weakness, decline, or simply old age. In the cramped stone houses along the Danube, families watched loved ones fade over months, first a cough, then the slow wasting that left cheeks hollow and skin stretched thin. Children who once played in the churchyard grew pale and silent, their laughter replaced by the hush of anxious prayers. Mothers sat by sickbeds through nights thick with fear, counting the ribs of a son who could no longer rise, or smoothing the hair of a daughter whose fever never broke. The household routine bent around illness. A father, once strong enough to haul sacks of flour to the market, now wrapped in blankets, stared at the ceiling, his breath shallow and rattling. Meals grew smaller as appetites vanished. Neighbors whispered about families struck by the same fate, yet no one spoke openly of the White Plague. The word consumption carried a quiet terror, too familiar to be named aloud, too common to be rare. Burial notes from the parish priest offered little comfort. One read, "Passed away from weakness." Another read, "Taken by decline." Behind these gentle phrases lay a truth that families suspected but could not prove. Some households lost child after child, while others watched a grandmother survive to 90, her body marked by scars of an old infection. The difference between survival and loss seemed random. A lottery played out in the shadows of each home. In the crypt, the evidence of these interrupted lives lay preserved in silence. Each coffin, each name, each faded ribbon told the story of a community shaped by invisible suffering. The true scale of the epidemic remained hidden, waiting for science to uncover what the living could only guess. Inside the sterile laboratory at University College London, every movement began with a checklist. Dr. Helen Donoghue and her team worked behind double-sealed doors, white coats zipped to the chin, sleeves snapped tight in latex. Ancient tissue, centuries old, carried not just secrets, but the risk of contamination at every step. Only a handful of researchers were allowed near the samples, each one trained to avoid even the smallest trace of modern DNA. The mummified ribs, selected for their dense bone and proximity to the lungs, arrived sealed in coated containers. Each was logged, weighed, and photographed before a single incision. Under a laminar flow hood, the surface of the bone was shaved away to remove any trace of outside contact. Tools were sterilized with ultraviolet light and bleach, then replaced after each use. The team spoke in low voices, aware that a single skin cell could ruin months of work. A fragment of rib, barely the length of a fingernail, was placed in a clean tube. The process to extract DNA from this material took days. The bone was ground to powder, then suspended in a solution designed to break down centuries of cross-linked proteins. The goal was to release any fragments of genetic material still trapped inside. The protocols had been refined over years to coax DNA from tissue that had survived 200 winters in the crypt's cold embrace. Polymerase chain reaction, PCR, was the next hurdle. This technology, standard in modern labs, was pushed to its limits here. Ancient DNA is fragile, broken into tiny pieces, often damaged by time. The team used primers designed to target the genetic signature of Mycobacterium tuberculosis, the bacterium responsible for the White Plague. Each reaction ran in triplicate, with negative controls at every stage to catch the faintest hint of contamination. Even a single false positive could cast doubt on the entire project. The first successful amplification sent a ripple through the lab. On the computer screen, a faint band marked the presence of tuberculosis DNA, preserved since the 18th century. More samples followed, each result carefully logged, cross-checked, and verified. The team's precision paid off. Dozens of mummies yielded genetic evidence of tuberculosis, their infections frozen in time. This molecular detective work, carried out in silence and under the strictest control, became the backbone of a new understanding, revealing a hidden epidemic that history had never recorded. While DNA analysis offered a breakthrough in tracing tuberculosis, through the centuries, another line of evidence brought its own kind of certainty. In a laboratory at the University of Szeged, Dr. Oksovsky Istvan Varadi and his team focused on the chemistry of the disease itself. Their approach relied on high-performance liquid chromatography, HPLC, to search for mycoserosic acids, rare lipid molecules produced only by Mycobacterium tuberculosis. Unlike fragile strands of DNA, these lipids endure. They cling to bone and tissue for centuries, outlasting even the most persistent genetic fragments. The process began with a sliver of mummy tissue, carefully weighed and dissolved in a solvent. The solution passed through the HPLC instrument, separating each molecule by size and chemical structure. Mycoserosic acids emerged as clear peaks on a digital readout, their presence confirming not just a possible infection, but a definitive fingerprint of tuberculosis. This chemical evidence became essential when DNA alone could not provide answers. In many cases, centuries had left genetic material too broken to amplify, or environmental microbes had overwhelmed the sample. Yet the lipids remained, stable and unmistakable. Even a single positive result could validate a suspected case, especially when matched with radiographic evidence of calcified lesions in the lungs or bones. The dual approach, genetic sequencing paired with lipid biomarker analysis, built an unshakable foundation for the research. It allowed scientists to reach deeper into the crypt's past, confirming tuberculosis in individuals whose DNA had faded with time. The method also opened new possibilities for mummy collections around the world, from Egypt to South America, where preservation conditions might differ, but the chemical traces of disease endure. For the VAC crypt, this chemical confirmation meant that every positive result stood on solid ground. The presence of mycoserosic acids, detected centuries after burial, left no doubt. Tuberculosis had swept through this community, its legacy written not only in the genes of the dead, but in the molecules that survive them. Numbers on a page rarely capture the weight of human suffering, but the data from the VAC crypt forced a reckoning. Out of 265 mummified bodies, 176 tested positive for Mycobacterium tuberculosis, an infection rate of 66.4%. In a single community, 2 out of every 3 people had carried the White Plague. The figures shattered old assumptions about tuberculosis as a sporadic visitor. Here it was a constant presence, woven into the fabric of daily life and death. The pattern did not spare the young or the old. Infants, children, adults in their prime, and elders in their 80s and 90s all showed evidence of infection. The disease did not discriminate by age or standing. In some family groups, burial records listed a grandmother and a grandchild only rows apart, both carrying the scars of tuberculosis. For 40.6% of those infected, the bacteria had not remained silent in the lungs. It had spread through the bloodstream, invading bones, organs, and joints, a sign of disseminated disease that often meant a slow, painful decline. Parish records told one story, deaths from weakness, decline, pneumonia, but the biological evidence told another. Many who died of supposedly unrelated causes were, in fact, consumed by tuberculosis. The mismatch between recorded cause of death and biological reality revealed how deeply the epidemic had been misunderstood. The crypt's population became a statistical portrait of an invisible catastrophe, exposing the true reach of a disease that shaped every household in Vac. The scale of infection, confirmed by modern science, reframed the history of tuberculosis in Europe. It was not a distant threat, nor a rare misfortune. It was an epidemic hiding in plain sight, touching nearly every family and leaving its mark on every generation. The numbers did not just count the dead, they mapped the silent advance of a killer that had haunted the community for more than a century. In a quiet corner of the University of Warwick, Dr. Gemma Kay and her team began to question what everyone thought they knew about tuberculosis. The standard belief was clear: one person, one strain. But as the sequencer's word and the digital readout stacked up, something unexpected appeared in the data. The DNA extracted from 8 mummified bodies in the Vac crypt did not tell a simple story. Instead, the fragments mapped to not just one, but multiple distinct strains of Mycobacterium tuberculosis inside the same individual. Most astonishing of all, one body contained genetic fingerprints from 3 different strains. In total, 14 unique TB genomes surfaced from just 8 individuals, each belonging to lineage 4, the same family of bacteria, still causing millions of cases across Europe and the Americas today. This was not a rare accident or a laboratory mistake. The team ran controls, checked for contamination, and repeated their sequencing. The result held firm. People in 18th century Hungary were being infected, reinfected, and co-infected by TB throughout their lives. The implications rippled through the research community. Modern patients almost never harbor more than one strain at a time. Yet here, in the sealed crypt of Vac, the dead carried evidence of a bacterial battlefield raging inside their bodies. The genomes even preserved ancestral forms of key genes, snapshots of the bacterium before the age of antibiotics, before resistance mutations began to sweep the globe. Dr. Kay's approach, using shotgun metagenomics, bypassed the need to culture living bacteria. It allowed the team to recover entire genomes from centuries-old tissue, capturing a moment when TB was evolving in real time within a single host. The discovery forced a new understanding. Tuberculosis did not move in simple, linear outbreaks. It surged through communities in complex, overlapping waves with individuals acting as crossroads for strain diversity. The Vac crypt had become a genetic archive, upending assumptions about how one of humanity's oldest killers spread and survived. Tuberculosis today is the deadliest infectious disease caused by a single organism. Each year, 1.3 million people die from a condition that, despite centuries of medical progress, remains stubbornly out of reach for a universal cure. The rise of drug-resistant strains has made treatment longer, harsher, and sometimes impossible. In some parts of the world, even the most powerful antibiotics fail, leaving doctors with few options and families with little hope. The Vac mummies, preserved by time and chance, now offer a rare opportunity to break this cycle. Researchers are preparing to compare the DNA of those who survived tuberculosis in the 18th century to those who did not. By searching for protective variants in immune system genes, scientists hope to uncover the biological secrets that allowed some people to control the infection without medicine. The plan is ambitious. Scientists will sequence the genomes of both survivors and victims, then match the findings against what is known about immune responses to tuberculosis today. If successful, these efforts could reveal new targets for vaccines or therapies, ways to mimic natural immunity that worked long before antibiotics were discovered. The genetic data from Vac may also help pinpoint vulnerabilities in the bacterium itself, especially in strains that have not yet developed resistance. For modern patients facing drug-resistant tuberculosis, the lessons hidden in centuries-old tissue could mean the difference between life and death. The urgency is clear. While history preserved these bodies in silence, the science they inspire is anything but quiet. Every new sequence, every tested hypothesis brings the world closer to treatments that could save millions. The story of Vac is no longer just a chapter in the history of disease. It is a blueprint for hope in a world still battling the White Plague. Drug-resistant tuberculosis now threatens millions, with current treatments losing ground every year. The genetic secrets preserved in these centuries-old remains could unlock new potential for defenses, if we choose to invest in the science. Humanity's next breakthrough may depend on lessons entombed for generations. The future of global health could rest on what we are willing to uncover from our past. What would you do to end an epidemic?